Hello, everyone. Thanks for joining my presentation today. I am Namhyeon Park. Today, I will talk about my project studying a potential food additive made of maltodextrin and fatty acid. To explain why I selected maltodextrin, I should start from one food additive, sucrose fatty acid ester. These compounds are approved by US FDA and widely used in the food industry because of their low impact on public health and the environment with useful functions. However, most sucrose fatty acid esters have low water solubility, resulting in limited application. To overcome this issue, I selected maltodextrin as a substrate. Among many different properties, I focused on its highly hydrophilic feature closely related to water solubility. Using a lipase, I synthesized maltodextrin fatty acid ester, and the synthesized compound was confirmed by HPLC and NMR. Also, the final compound showed good water solubility. So using the synthesized compound, I studied its ability in an emulsion system. Firstly, I prepared 20% oil in water emulsion containing compound, and the increasing clarification layer was tracked over storage to evaluate emulsion stability. Here, I used one standard that a stable emulsion has less than 1 millimeter of clarification layer per day. So any lines below this standard line mean stable. Based on this, maltodextrin fatty acid ester stabilized emulsions at two different storage temperatures. However, sucrose fatty acid ester could not stabilize emulsions at the same condition. Then, I studied droplet sizes of emulsions to analyze an emulsion forming ability. Here I used another standard about preferable emulsions having less than 1 micrometer size of droplets and consistency during storage. Fresh emulsions containing maltodextrin fatty acid ester showed broad droplet distribution, and most droplets were larger than 1 micrometer. And even after storage, these emulsions were shifted to generally more droplets less than 1 micrometer, but bimodal distribution indicating not stable emulsions. However, sucrose fatty acid ester produced emulsions having narrow droplet distribution around 1 micrometer size, and these emulsions were stationary over storage, indicating stable emulsions. These results indicate the enhanced emulsion stabilizing ability of maltodextrin fatty acid ester, even though it has a lowered emulsion forming ability. To explain the results, I suggested one hypothesized mechanism. Sucrose fatty acid ester has a balanced hydrophilic and hydrophobic property with a small size, and those properties made this compound quickly absorbed into the oil and the water interface during emulsion formation, producing stable oil droplets. However, maltodextrin fatty acid ester may not rapidly participate in emulsion formation because of its large size and unbalanced hydrophilic hydrophobic property. Instead, the large maltodextrin part may act as a stabilizing layer, increasing emulsion stability. Then I studied in vitro microbial inhibitory ability. Briefly, maltodextrin fatty acid ester inhibited all 11 microorganisms tested, but sucrose fatty acid ester could not inhibit 3 microorganisms, indicating the broader inhibitory spectrum of maltodextrin fatty acid ester. Then, to better understand antimicrobial actions, I studied the relationship of growth inhibition with cell morphology and membrane leakage, which are related to cell membranes. These are the microscopic images of different cells. The pattern of morphological changes corresponded to the inhibition pattern, indicating a possible inhibitory action of cell membranes. Then I measured extracellular proteins as an indicator of cell membrane leakage. Here, the increased extracellular protein pattern corresponded to the cell inhibition pattern, except geobacillus cells, proposing a partial relationship between cell inhibition and the altered membrane leakage. Based on my results and previous literature, I suggested the possible inhibitory mechanism. Both compounds may mainly target cell membranes, and on this occasion, compounds can insert into cell membranes, increasing membrane leakage, or these compounds can interact with other membrane-related components. Notably, during the insertion, maltodextrin fatty acid ester may deliver enhanced steric disturbance on cell membranes, leading to the broader inhibitory spectrum. Then I also tested my compound in different food models other than growth media. In short, organic acid in foods could synergistically act with my compound to attack cell membranes. High levels of fat, starches, and oils could interrupt the approaches of my compound to cell membranes, decreasing inhibitory action. In the case of the temperature, its effect was more complex, probably because the temperature can affect both compounds and bacterial cell membrane status. Lastly, to evaluate as a food additive, I studied in vitro cytotoxicity of maltodextrin fatty acid ester against 2 different human intestinal cell lines. Firstly, sucrose fatty acid ester showed high toxicity in the ranges of concentration tested. However, maltodextrin fatty acid ester showed non-toxicity, suggesting the lowered human intestinal cytotoxicity. In conclusion, enzymatically synthesized maltodextrin fatty acid ester can be a potential safe food additive. Thank you.